There exists a biological mystery that has puzzled geneticists, historians, and physicians for nearly a century. It is not a disease, not a curse, and certainly not something that was understood before the modern era of medicine. What we are discussing is a genetic variation so unusual, so statistically improbable, that its very existence challenges our fundamental understanding of human evolution and adaptation. This is the story of Ih-negative blood, a condition that affects approximately 15% of the global population, yet remains one of the least understood biological phenomena in contemporary science. When we think about what makes us human, we often think about our DNA, our capacity for language, our ability to create complex societies. We rarely think about what flows through our veins. Blood has been romanticized throughout human history as the essence of life itself, the carrier of our vigor, our lineage, and our identity. In medieval times, blood type meant nothing because the science did not exist to measure it. Yet in our modern age, where we can sequence entire genomes and peer into the molecular machinery of cells, we still find ourselves confronted with questions that remain unanswered. Why does a significant portion of humanity carry a blood type that defies the normal rules of evolution? Where did it come from? And perhaps most intriguingly, what advantages or disadvantages does it confer? The story of Ih-negative blood begins not with a discovery of something new, but rather the discovery of something missing. In the 1930s and 1940s, medical researchers began systematically studying blood types, building on the earlier work that had identified the major blood groups. What they found was startling. While some people's blood contained a specific protein, which they would eventually name the rhesus factor, or Rh factor, named after the rhesus monkey used in the original research, other people's blood completely lacked this protein. These Rh-negative individuals represented a mystery within a mystery, because this protein, this marker, appeared to be present in virtually every other primate species on Earth. The very animals from which we are supposed to have evolved all possess this protein, all except for a percentage of modern humans. From an evolutionary perspective, this should not exist. The fundamental principle of natural selection dictates that traits which persist across an entire species lineage remain because they provide some survival advantage or at minimum do not provide a disadvantage significant enough to be eliminated. We see this principle play out across the entire animal kingdom. If a characteristic is universal among primates, from chimpanzees to gorillas to the smallest lemurs, then that characteristic should also be universal among humans, who are themselves primates. Yet here we have a trait that appears to be entirely absent in our closest living relatives, while remaining present in a small but significant portion of the human population. Some evolutionary biologists have proposed various theories to explain this paradox. One hypothesis suggests that the loss of the Rh factor occurred relatively recently in human evolutionary history, perhaps within the last 10 to 20,000 years, through a process known as genetic drift. According to this theory, in certain isolated populations, a random mutation that deleted the gene responsible for producing the Rh protein became common not because it was advantageous, but simply because of chance. In small, isolated communities, random genetic variations can become established at much higher frequencies than would be predicted by the normal rules of probability. This phenomenon has been observed in numerous human populations around the world. However, this explanation alone does not fully account for the presence of Rh-negative blood today. According to mathematical models of population genetics, if Rh-negative blood truly carried no selective advantage and provided some disadvantage, as the historical evidence of complications during pregnancy suggested, then the frequency of this blood type should have declined over time, eventually becoming extinct. Yet it has persisted. It has remained stable, and in certain regions, it has even increased in frequency as modern medicine has solved the reproductive complications that once made it potentially lethal. Another perspective comes from the study of ancient human populations. Some researchers have proposed that Rh-negative blood may be a remnant of earlier human groups who inhabited Europe before the arrival of populations from Africa and Asia Minor. If true, This would mean that the Rh-negative individuals represent a continuation of a more ancient human lineage, a thread of genetic continuity stretching back thousands of years. The evidence for this theory comes partly from geographic distribution patterns. Rh-negative blood is far more common in Europe, particularly in Northern and Western Europe, than it is in Africa, Asia, or among indigenous populations of the Americas. This distribution pattern is consistent with the hypothesis of an older, geographically bound population. When scientists began mapping the distribution of Rh-negative blood across the globe, an unusual pattern emerged. The highest concentrations were not randomly scattered but clustered in very specific regions, particularly in Europe. Most striking was the concentration of Rh-negative blood in the Basque population of northern Spain and southwestern France. In parts of the Basque country, the frequency of Rh-negative blood reaches as high as 35 to 40%, more than double the global average. The Basque people themselves represent an enigma within European populations. They speak a language, euskera, that is linguistically unrelated to the major language families of Europe. They have maintained a distinct culture and identity despite centuries of political pressure to assimilate into surrounding Spanish and French societies. Their genetic profile is distinctive, showing less admixture with other European populations than would be expected given their geographic location. Beyond the Basque region, elevated frequencies of Rh-negative blood are found in Celtic populations of Ireland, Scotland, and Wales. The Atlantic coastal regions of Portugal and Brittany show higher frequencies than inland areas. This pattern has led some researchers to propose what they call the Celtic connection, the idea that populations that remained relatively isolated along the Atlantic coast of Europe preserved higher concentrations of Rh-negative blood than populations that experienced more migration and genetic mixing with incoming groups from the Mediterranean and Central Asia. The geographic distribution becomes even more intriguing when we consider population movements and migrations throughout human history. The areas with the highest Rh-negative frequencies are precisely the regions that would have been most geographically isolated before modern transportation technology made easy movement possible. Mountains, islands, and coastal regions protected by treacherous waters would have naturally limited gene flow with surrounding populations. In such isolated communities, any genetic variant present in the founding population will be preserved at higher frequencies than in more cosmopolitan areas. This genetic isolation could explain the puzzling persistence of Rh-negative blood in these regions. For centuries, before modern medicine, Rh-negative blood carried a terrible cost. The problem arose specifically in pregnancy. When an Rh-negative woman carried a baby who had inherited the Rh-positive blood type from the father, her immune system could potentially recognize the baby's blood as foreign. During delivery, fetal blood cells could enter the maternal circulation. The mother's immune system, finding these cells strange and dangerous, will begin producing antibodies against the Rh antigen. This sensitization during the first pregnancy was usually not catastrophic. The first child often survived. However, in subsequent pregnancies, the mother's pre-existing antibodies could cross the placenta and attack the fetal red blood cells, causing a condition known as hemolytic disease of the newborn. This could result in severe anemia, jaundice, brain damage, or fetal death in utero. Before treatment was available, Rh-negative women faced a heartbreaking choice: either limit themselves to one child or accept the risk that any subsequent children would be born severely ill or stillborn. This represented a profound evolutionary disadvantage. In a historical context where infant mortality was high and families needed many children to ensure that some would survive to adulthood, having a blood type that effectively rendered you unable to bear more than one healthy child should have been selected against with tremendous force. Yet somehow, the Rh-negative blood type persisted. The breakthrough came in 1968 with the development of RHIG, a medication commonly known as ROGAM. This treatment works by introducing a controlled amount of antibodies against the Rh antigen into an Rh-negative mother's bloodstream during or immediately after delivery. These antibodies bind to any fetal red blood cells that have entered the maternal circulation, marking them for destruction before the mother's immune system can encounter them and become sensitized. This simple but elegant solution essentially prevented the immune sensitization that would have caused problems in future pregnancies. Suddenly, Rh-negative women could have as many children as they wished. One of the barriers to reproduction that had limited Rh-negative individuals for thousands of years was effectively eliminated. What happened next was as interesting as the medical breakthrough itself. Once the reproductive disadvantage was removed, the frequency of Rh-negative blood in the population did not remain stable. It began to increase. This increase was particularly marked in developed nations with access to modern prenatal care and RHIG treatment. Within a single generation, the balance shifted. What had been a genetic liability became simply a different variant with no particular disadvantage compared to Rh-positive blood. This change in the selective landscape has profound implications for how we think about genetic variations and human evolution. But what if the story of Rh-negative blood is not simply one of a genetic neutral variant that happened to persist? What if there are advantages that compensate for the reproductive disadvantage? Advantages so subtle or so specialized that they only become apparent when we look carefully at the patterns of human history. One intriguing hypothesis comes from studying infectious disease. Many of humanity's most dangerous pathogens, including bacteria and viruses responsible for plague, smallpox, and other historical killers, attach to red blood cells or blood proteins to gain entry into cells or to evade the immune system. The Rh antigen might serve as one of these attachment points. If an individual lacks the Rh antigen, certain pathogens might have a harder time establishing infection. During times of epidemic disease, and human history has seen many such times, this could represent a genuine survival advantage. The evidence for this hypothesis remains suggestive rather than definitive. Some historical researchers have noted that during the Great Plague epidemics that swept through Europe in the Middle Ages, certain populations appeared to have better survival rates than others in the same geographic regions. While many factors could explain such variations, including differences in population density, trade patterns, and social behaviors, it is plausible that genetic factors played a role. If Rh-negative individuals were more resistant to plague, they would have been more likely to survive to reproductive age and pass on their genes. Over multiple generations, such an advantage could maintain and even increase the frequency of Rh-negative blood in a population. Another potential advantage lies in resistance to parasitic infection. Some research has suggested that Rh-negative individuals may have greater resistance to toxoplasmosis, a parasitic infection spread by cats that affects a significant portion of the world's population. If true, this could represent another subtle but consistent advantage that might accumulate over time. Beyond the medical and evolutionary perspectives, Rh-negative blood has attracted considerable speculation about more ancient or unusual origins. Some researchers have proposed that the Rh-negative trait may represent genetic influence from archaic human species such as Neanderthals, who shared Europe with early modern humans and left traces of their DNA in modern European populations. The idea is that Neanderthals, having evolved separately in Europe over hundreds of thousands of years, may have possessed different blood type systems. When modern humans arrived in Europe and interbred with Neanderthals, they may have inherited genetic variations including modifications to the Rh system. This hypothesis remains highly speculative, and the genetic evidence does not currently support a direct Neanderthal origin for Rh-negative blood. However, it demonstrates how scientists attempt to integrate various lines of evidence to understand unusual genetic phenomena. The geographic and cultural isolation of populations with high Rh-negative frequencies has led to other speculations. Some researchers have wondered whether the linguistic isolation of the Basque people, who speak a language unrelated to neighboring European languages, and their genetic isolation might be related to deeper historical roots. Could the Basque represent a remnant of a much more ancient European population than previously thought? The evidence remains ambiguous, though genetic studies do suggest that Basque populations have a longer continuity in their geographic region than most other European populations. Today, the most important practical significance of Rh-negative blood lies in its role as the universal blood type. O-negative blood, which lacks both the A and B antigens as well as the Rh antigen, can theoretically be transfused to recipients of any blood type. This makes O-negative blood extraordinarily valuable in emergency medicine. When a trauma patient arrives at a hospital in critical condition, often there is no time for blood typing. Doctors must act immediately to prevent death from blood loss. In such situations, O-negative blood is the only choice, because it is the only blood type that will not trigger an immune reaction in recipients with any other blood type. Because O-negative blood is so universally useful, hospitals maintain large stocks of it for emergencies. However, only about 6 to 7% of the global population is O-negative. The demand for this blood far exceeds the natural supply from voluntary donations. This has created a chronic shortage of O-negative blood in many parts of the world. Blood banks actively recruit O-negative donors and often offer incentives to encourage them to donate regularly. For individuals with O-negative blood, donating has become recognized as one of the most direct ways to save lives, potentially allowing them to help dozens of people with a single donation. This medical reality has shaped how Rh-negative individuals think about their blood type in the modern world. What was once a source of reproductive anxiety is now increasingly viewed as a gift to humanity. Public health campaigns in many countries specifically target O-negative donors, emphasizing the critical importance of their blood for trauma care and emergency medicine. The shift in perspective from liability to asset represents one of the most dramatic changes in how a genetic variant is perceived within a single lifetime. Within communities of Rh-negative individuals, certain traits and characteristics are frequently reported as being associated with the blood type. These include a tendency toward red or blonde hair and a higher frequency of light-colored eyes. Some Rh-negative individuals report a lower baseline body temperature, often running slightly cooler than the population average. Others claim heightened sensitivity to electromagnetic fields, unusual emotional sensitivity or empathy, or an affinity for intuitive thinking. It is important to note that most of these claimed associations have not been rigorously proven through large-scale scientific studies. Many are based on self-reported data, community observations, or anecdotal evidence. This does not mean they are false, but rather that they remain in a category of interesting observations that await more systematic investigation. Some of these traits might represent actual genetic correlations, while others might reflect confirmation bias. People with Rh-negative blood noticing and remembering instances where they exemplify the trait they expect to have. Still others might reflect real patterns in certain populations that are specifically studied, without being universal to all Rh-negative individuals. Nevertheless, the fact that thousands of people who share a rare genetic marker report similar traits and experiences is itself noteworthy. Whether these reflect genetic reality, cultural identity, or some combination of both, they have created a sense of shared community among Rh-negative individuals. Online forums, social media groups, and community organizations dedicated to Rh-negative individuals